I'm Dr. Ronald Berkman. This particular video will be discussing the structure and mechanisms of action of hormonal contraceptives. By viewing the videos that are in this particular segment for your education and reading the assigned textbook chapter, as well as attending the case discussion which will follow, participants should be able to describe all of the elements listed here. This particular video will cover that which is listed here in the sort of the yellow or gold coloring, and that is we will be discussing major structural features of contraceptive hormones, their mechanisms of action, and some of the challenges in formulating contraceptive hormones. The challenges are as follows. Certainly in the 1930s it was first established through animal studies that it looked like progesterone could inhibit ovulation. However, it was also noted that most naturally occurring reproductive hormones were not active if taken orally, which obviously would limit their use in patients because there would not be an easy mode of delivery. However, research in the 1950s did lead to the development of derivatives that were active orally, both of estrogen and of progestin, and began with the development of the first oral contraceptive for women. And approval of this contraceptive was in 1960 through the Food and Drug Administration. And since then, certainly there's been a lot of challenges and changes that have gone on. And the challenges have been to improve both the efficacy and to reduce side effects and serious sequelae, and also to explore and develop additional delivery systems other than the oral route. Certainly, to have a full understanding of contraceptive steroids, you should have a little bit of knowledge about their conversion from cholesterol. The process basically involves cleaving off its side chain. Then a variety of enzymes make structural changes, which again lead to progesterone, which in turn can lead to development of androgens, which then through additional enzymatic activity will in turn develop into progestins. Certainly changes in the progestins also will lead to additional potent progestins, although currently there is only one potent estrogen that's utilized in the medications now for contraception. And certainly all of these additional structural changes are in an effort to improve their potency, but also to reduce side effects and serious sequelae. So we start out with the typical steroid nucleus or structure with the 4 rings and the labeling of the carbon atoms as noted here, something I'm sure many of you have some recollection to a varying degree from your biochemistry days. As we follow the pathway, you see cholesterol in the upper left-hand corner, which through a hydroxylase will go to pregnenolone, and in turn through a dehydrogenase enzyme, pregnenolone can go to progesterone. Then these sort of follow south to hydroxypregnenolone and hydroxyprogesterone after being acted on by 17-alpha hydroxylase. And then as we go to the next slide, we pick them up again, but now they're being activated through 17-20-desmolase to dehydroepiandrosterone, which is DHEA, which also can be converted to androstenedione through a dehydrogenase process. These then go on to a testosterone through the pathways illustrated here. Through aromatase, these androgens also can form estrogens, as also shown here. So again, they're all somewhat interrelated. How important it is to know exactly all the structures. And the enzymes, again, is not overly important, but you should have some general knowledge of why these work the way they do. It's also useful in terms of trying to identify steroid structures if you're not certain as to how many carbon atoms are in the particular molecule being demonstrated. Progesterone tends to have 21 carbons. Estrogen 18 and testosterone 19. But again, with many of these large side chains that are in some of the newer developments, sometimes it's a little difficult to identify what the structure really represents. We start out first with estradiol. This is the most potent estrogen that is occurring naturally. You can see how the 4 rings are laid out, as well as the beginning of the side chains. Through insertion of an ethinyl group at the alpha position at C17, this is the basic change of estradiol that leads to the potent oral ethanoestradiol, which is contained in all of the contraceptive products currently that utilize estrogen. And you can see here's where that chain has been attached at C17. When you look at progestins, there's a whole host of progestins that are utilized in oral contraceptives, but also in alternative delivery systems such as the implant and the IUD. First generation ones were derived directly from testosterone. They include norethindrone, norethindril, and the others listed here. They were labeled estranes, pregnanes, which were somewhat different, and they include medroxyprogesterone acetate, the progestin that's in Depo-Provera. Second generation development led to gonanes from testosterone, included particularly levonorgestrel. Then came the third generation of progestins, which are derivatives primarily of levonorgestrel, and these included desogestrel, gestodine, norgestimate, and through modification of desogestrel, etonorgestrel. And finally, we have the fourth generation progestins, which have structures that are dramatically different than the rest, and these include drospirenone as well as nesterone and nomegestrol, which are not utilized much in this country. So we start out with progesterone, which is the naturally occurring progestin. But in reality, when we look at the structures of the progestins that are used in the various contraceptive products, they much more commonly look like testosterone as their derivative. And as we will point out, that's why some of these particular compounds are labeled nortestosterone in their nomenclature. So by modifying testosterone in the following fashion, that is attaching an ethinyl group at C17 and removing the C19 carbon, all of this leads to more progestational activity and some reduction in androgenicity. But again, removal of the C19 leads to many of these compounds that have C19 missing as being 19-nortestosterone derivatives. And we can see here's where that ethinyl group has been attached at C17. And you can see when you look at 19, there's no longer a carbon there, but there's just a hydrogen atom. Modification of testosterone to levonorgestrel does some of the same things, but now there's also an additional attachment of an ethyl group at C13, which appears to increase the overall potency. But again, this is coupled with maybe some increase in androgenicity. And we can see again here at C13, sort of between the C19 and C17, where that group has been added. Modification of testosterone to norgestimate leads to additional requirements for changes in the structure. You still have the ethinyl group at C17, removal of the carbon at C19, as well as an attachment of an ethyl group at C13, and an additional attachment of an oxime group at C3. The attachment at C3 is felt to reduce androgenicity in these particular compounds. And here you can see, particularly well over to the left, the compound with the nitrogen is the attachment we just discussed. Note that there's sort of two versions of this compound, and when you look at norgestimate, it actually has equal amounts of this whereas if you call the drug norgestromin, it will include only one of the variants. And again, this changes the stoichiochemistry of how this particularly looks if you sort of look at it structurally in that way, in a three-dimensional fashion. And then we have desogestrel, where again modifications of it will lead to etonorgestrel. And this is done by, again, the C17 and C19 modification we've been talking about, but now with the attachment of a ketone at C3. And you can see down here where we had with norgestimate, the oxine group, here we have the ketone attached. And etonorgestrel, as I think most of you are aware, is the progestin that's in the contraceptive implant. And finally, we have sort of these fourth generation compounds such as drospirenone, which basically is certainly odd in terms of its look, but also seems to have a lot of activity that reduces androgenicity because it appears to be derived similar to spironolactone. And we also have medroxyprogesterone acetate, which actually is one of the earlier forms of progestins, but clearly is far different from others. It certainly is not a C19 nor testosterone derivative. And again, this is the drug that's in Depo-Provera. So how do these work in terms of causing their activity or actions as contraceptives? The estrogens can lead to central inhibition of GnRH, leading to some suppression of FSH. They also increase secretory effects on the endometrium, which reduces some likelihood of implantation, but also stabilizes endometrium to reduce breakthrough bleeding. So again, it's important estrogen in some of the earlier compounds was at a much higher dose, but also breakthrough bleeding was far less than what we encounter nowadays. It also interferes with the normal lifespan of the corpus luteum, but given by itself, it is less effective in inhibiting ovulation than the progestin that's usually added to the estrogen. And certainly the progestins have hypothalamic activity that reduces mid-cycle surges of LH so that ovulation doesn't occur. Progestins also will suppress normal development of endometrium, leading to pseudodecidualization of the stroma and narrow glands, which reduce likelihood of implantation. And also cervical mucus becomes very thick that interferes with sperm ascent. And again, it's felt, for example, in some medications like medroxyprogesterone, which is given in the Depo form. Patients still can occasionally ovulate while on this, and yet the drug may be active, and it may be this latter effect that makes their contraceptive efficacy persist. So with that in mind, what are the persistent challenges that we are facing in hormonal contraception? Certainly, as the estrogen dose has been lowered, and the reason for lowering it is to reduce the risk of venous thromboembolism, particularly in oral contraceptives, this has led to more breakthrough bleeding. And so what manufacturers have done has been varying hormonal doses at various parts of the pill cycle in attempts to address this. And these is where the so-called triphasic pills have come in, where again dosing varies with an idea that this hopefully will reduce some of the troublesome side effects. The other challenge is having a potent enough progestin that at a low dose has good contraceptive properties, but this dose where it is good as a contraception is below the threshold of the dose response, for example, for a side effect or a serious sequelae. Also, increasing potency, particularly of progestins without increasing side effects, reduces the volume of drug needed so that when it's put in slow delivery system, such as the levonorgestrel IUD, it increases to have products with longer times of effectiveness. For example, since progesterone itself is far less potent than, let's say, levonorgestrel, one could not, for example, have a IUD device that contain progesterone because it would be far too large and not practical, particularly if you're trying to have a product that lasts for 3 to 5 years or potentially longer. Here's what we mean by having a more potent progestin in the dose to reduce side effects. If we look at the red dose response curves, which there are 2 of them, and then the dotted line yellow dose response curves, and assume again the progestin dose listed on the x-axis varies between those 2 components, you can see that if we assume that the left or the initial dose response curve in the red is the one that leads to the contraceptive protection, that is, reduces ovulation, etc., and the second red dose response curve is a side effect such as breakthrough bleeding, or could be something more serious such as venous thromboembolism, you can see that these dose response curves overlap, meaning that with normal dosing a particular side effect may occur. If we now looked at the dotted yellow dose response curves and assume that the one on the left is the dose response curve required for contraception, but is at a potent enough dose at a lower dose, such that when you then look at the side effect, which is the second dose response curve, you can see they are separated such that there's no overlap, and so that in the usual dosage required for contraception, it is unlikely that the side effect is going to occur. The final thing to talk about a little bit about pharmacokinetics is how the various delivery systems sort of behave. And here is actual data from 2 transdermal patches and an oral contraceptives. If we look at the red dotted line, you can see the typical dose response curve of an oral drug. You have a rapid climb to its peak, and then slowly sort of it declines, and then the next pill is given the following day, as it is for a birth control pill, and you have the typical dose response curve, let's say over a week's period of time. Then you look at the top dotted green curve, and this is actual curve plotted for Ortho Evra, which was the initial contraceptive patch. And here you put the patch on at time 0, slowly rises to the level that you see here for ethanoestradiol, persists and starts falling off around day 7 until the next patch is put on. Then you look in the blue is the dose response curve for a new patch that actually just has been approved, which I'll mention a little more in a second. Here, because you have a more potent progestin and it's felt that you did not need as much estrogen, you can see that the patch, when this one is put on, the levels of ethanoestradiol are much lower than the levels of ethanoestradiol that one sees with the earlier developed patch. And the question is, can these pharmacokinetics of the pill, these transdermal patches, lead one to predict side effects? And certainly the way these analyses are done are so-called areas under the curve. Through computation, one can determine what the total amount of, say, an estrogen is given when you give an oral contraceptive with its dose response curves, which again vary each day, versus, let's say, a patch. And when they did the analysis for the initial patch, which was Ortho Evra, which now is being marketed as Xulane, there was 60% more ethanoestradiol being delivered to users in a contraceptive cycle compared to an oral contraceptive user using the same hormones. And the concern obviously is that the risk of venous thromboembolism appears to be associated with estrogen use and may be obviously worse with higher doses. Also keep in mind that estrogen is still used to stabilize the endometrium to reduce undesired bleeding. So a number of studies have been done to try to determine, is there a substantial difference in the risk of venous thromboembolism with the original patch, and there is some increase in the absolute risk of venous thromboembolism, but in general it appears that this risk is modest and certainly has not been enough that the medication or delivery system has been removed from use by the Food and Drug Administration. Now the newer patch, which is going to be marketed as Twerla, contains levonorgestrel as the progestin, not norgestimate. And it, as shown, provides substantially less estrogen per cycle, though a calculation I've not seen between the two. But even with this much lower estrogen being delivered per cycle, in the original studies that led to approval of this drug, there were 4 cases of venous thromboembolism out of the 1,735 women studied. But these all occurred in women with BMIs greater than 30. So this may have been effect that affects only obese women. But again, the total number of women studied, the number of cycle studies and so forth is inadequate to fully characterize the risk of venous thromboembolism, such that larger studies after the medication has been available and there's been more users will be needed to better characterize the risk of venous thromboembolism with this new patch. Relative to abnormal bleeding, it's of interest that after 6 months of use among the users of the original patch, only 6 to 7% of users had complaints of abnormal or breakthrough bleeding, whereas in the initial studies of the newer patch, the one that just been approved, 42% of women had complaints of breakthrough bleeding, albeit for short periods of time. So the bottom line is, although pharmacokinetics look interesting, they have not yet consistently predicted risk of adverse outcomes or side effects in a way that pharmacokinetics can be used clinically to any great extent. So in summary, what I've tried to do in this particular discussion is review much of how contraceptive hormones evolve. Certainly they continue to evolve. The alteration in their chemical structure certainly does influence potency and risk of side effects. Increasing potency, decreasing side effects, improving safety certainly is a large goal of drug development, as is expanding delivery systems that are easy to use, effective, and also have acceptable side effects.